Hello, this is Gina Piscatelli with the 6th lecture on the muscular system. And in this lecture, we're going to look at the coupling between excitation of a muscle cell and its contraction. And then we'll finally look at how a muscle relaxes or what needs to happen for relaxation to occur. So let's start off with excitation. You're familiar with this picture. It shows you a neuromuscular junction, and acetylcholine is released by the neuron and binds to a receptor on the motor end plate of a muscle cell. That causes potassium to leave and sodium to enter, but a lot more sodium enters than potassium leaves, so the inside of the cell becomes depolarized. Now, if depolarization reaches threshold, which we'll talk about in a second, then some other channels open and close on the neighboring side here of the membrane. And those will be called voltage-gated channels. And they are ion channels, just like in a neuron. So what will happen is an action potential will occur in the muscle cell, not in a neuron, but in a muscle cell. So an action potential consists of a muscle cell starting at resting, which is about negative 90 millivolts instead of negative 70. And that influx of sodium reaches threshold, which is approximately negative 65 millivolts. And that opens up voltage-gated sodium channels. So now more sodium goes into the muscle cell. Those voltage-gated sodium channels stay open until about 30 or 35 millivolts, and then they close. So sodium channels close at that voltage. Then potassium channels open at the same voltage, and now potassium leaves the muscle cell, and the muscle cell repolarizes in the sense that it becomes more negative. And it drops down to resting, and there is a corrective period where the sodium-potassium pump has to fix the concentration of sodium and potassium in versus out of the cell. So an action potential occurs in response to the motor end plate being depolarized. And during depolarization, we have sodium channels that are open, so sodium goes in. But then during repolarization, potassium channels are open, and so potassium leaves. Now, how is that coupled to contraction? That's just excitation. What happens is the end plate has become depolarized due to the neurotransmitter and sodium ions, and action potentials occur along the length of the cell membrane, just like in a neuron. But remember, the cell membrane has these deep extensions that we call T-tubules or transverse tubules, and so the action potentials move deep into the muscle cell. This depolarization causes calcium channels to open in the terminal cisterna. So the terminal cisterna, right adjacent to the T-tubule, has calcium channels that sense that change in voltage, and they open. So now calcium is released from the sarcoplasmic reticulum and goes out into the cytoplasm where the myofilaments are. So when calcium is present in the cytoplasm, we can get crossbridge formation. And when we get crossbridge formation, we know that myofibrils and sarcomeres shorten, and therefore a whole muscle will shorten. So the steps of excitation contraction are, first we have to have a neurotransmitter released from a neuron, and then we get sodium influx into the muscle cell. Then, when the motor end plate is depolarized to threshold, some more sodium channels open, and that generates an action potential in the muscle. So this is all the excitation. Muscle cell is excited. And there's a wave of action potentials that travel to the T-tubules, which causes the calcium channels of the sarcoplasmic reticulum, the terminal cisterna, to open. Now, once calcium channels are open, calcium leaves the sarcoplasmic reticulum, and now we have calcium for the next part, the contraction part. You need calcium for contraction, because calcium is going to bind to troponin. When troponin binds to calcium, it shifts, and that causes tropomyosin to shift. And now the myosin binding sites on actin are exposed, and actin-myosin crossbridges form, and the muscle contracts. So overall, if we add it together, we had to have a neuron that was excited for sure. But that translated into excitation of the muscle cell and release of calcium from the sarcoplasmic reticulum. Once there's calcium, now you can get contraction. And so the muscle shortens due to contraction. Now, what needs to happen for that muscle to stop contracting or to relax? Well, first we need to stop the acetylcholine release, right? So the neuron stimulation needs to stop. And that's going to be somewhere in the brain and the central nervous system and then the peripheral nervous system. But in addition, in the synaptic cleft, there is an enzyme, or at the neuromuscular junction, there's an enzyme called acetylcholine esterase that breaks down acetylcholine. So back at the neuromuscular junction, acetylcholine, some of it will diffuse out of the cleft. That diminishes how much is present there. And some of it is broken down by this enzyme called acetylcholine esterase. Then, in addition, we need to have ATP around so that myosin will bind to ATP and release the actin to dissociate the cross bridges. And calcium needs to be released from troponin so that it can be put back in the sarcoplasmic reticulum, and the myosin binding sites on actin have to be covered up again. So we have to stop the neuron. We have to diminish how much acetylcholine there is. Some of that's from stopping the neuron. We need ATP around. And we need to sequester that calcium. Those are the big ones. Now, I thought I'd mention a little bit about rigor mortis here. I mentioned it briefly in an earlier lecture, but rigor mortis is defined as hardening of the muscles and stiffening of the body. It happens after death, 3 to 4 hours. And it occurs because any myosin heads that are bound to ATP will be breaking down ATP into ADP and inorganic phosphate, which of course allows the myosin head to bind to actin. No energy required, no oxygen required, no nutrient required, so death doesn't matter. So you'll still get cross bridges forming. And this process peaks about 12 hours after death, and it lasts, I don't know if this is accurate, but it lasts for maybe a day, right? So it diminishes and disappears somewhere between 2.5 days, 2.5 days. Now, another thing that causes the formation of actin-myosin cross bridges is not just the fact that any leftover ATP has been broken down, but also there's calcium released from the sarcoplasmic reticulum. Why is that happening? It's happening because death has occurred and cells are deteriorating, and so the sarcoplasmic membranes, sarcoplasmic reticulum membranes breaks down, and calcium is just dumped into the cytoplasm. It's unregulated. So while you've got myosin without ATP and lots of calcium from the breakdown of the cell, you're going to get cross-bridge formation. And if the cells aren't alive and taking in nutrients and oxygen, you don't have any more ATP production, so the cross bridges can't detach or release, and you can't get any muscle relaxation, like normal regulated muscle relaxation. Now, of course, these muscle cells continue to deteriorate to the point where the myofilaments even break down. And of course, that will mean that the cross bridges are broken, and then the muscle won't be so stiff anymore because it won't be contracted, mainly because the proteins inside aren't even intact anymore. So that's what rigor mortis is. Okay, that is the end of lectures for week 14, and we have 2 more for week 15.